Our today's topic is Ephedra, and in this class we'll be discussing its morphology and anatomy. Ephedra belongs to class Nitopsida, order Ephedrales, and family Ephedraceae. Family Ephedraceae is a monotypic family, that is with only one genus, Ephedra, which comprises 40 different species. Ephedra is a small, much branched, you can see here, this is Ephedra plant, and Ephedra is a small, much branched, erect, procumbent, or occasionally a climbing shrub. It is distributed chiefly in the arid regions of the world. A widely used alkaloid, ephedrine, is extracted from some of its species. Next is distribution and habitat. As I already told you, genus Ephedra has about 40 different species that are distributed in the arid regions of New and Old Worlds, from Mediterranean and Black Sea shores up to China, including northern and northwestern India. The Old World means France, Canary Islands around the Mediterranean east to Iran, India, and China accounts for about 18 species, whereas roughly 22 species are confined to the New World, that is North and South America. The plants exhibit extreme xerophytic characters and are mostly shrubby, generally having a height of less than 2 meters. However, some species are lianas, others spread by underground rhizomes, and one species, Ephedra preiandra, is a small tree whose stem has a diameter of about 30 centimeters. On a casual look, one may mistake a plant of Ephedra for Equisetum. Equisetum is a tridophyte, as its internodes are longitudinally ridged, and also ridges at successive internodes alternate. Similarly, the leaves on successive internodes also alternate. There are two types of branches present in Ephedra, indeterminate and determinate branches, but the distinction not as well marked. The indeterminate branches at each node has leaves in whorls of threes or sometime in fours. The determinate branches are borne in the axil of the leaves of some indeterminate branches, and these bear opposite and decussate leaves. Now we'll discuss morphology and anatomy of Ephedra. So first of all, we'll start with the stem. Young stem. As mentioned earlier, the longitudinal ridges on the stem impart it a wavy outline. You can see here, this is Ephedra plant. This is young stem of Ephedra. On the nodes, reduced scaly leaves are present. In transverse section, this is transverse section of young stem of Ephedra. This, due to the presence of longitudinal ridges on the stem, its outline looks wavy. It shows the presence of ridges and furrows. The epidermis, outer layer, is epidermis, and epidermis has a thick cuticle which is interrupted in furrows. As you can see here, look at this diagram. In the furrows where stomata are present, and the stomata are sunken, and each consists of two guard cells with a prominent substomatal cavity. Next part is hypodermis, and hypodermis is present only below the ridges, right? Here, this is hypodermis, and is composed of elongated sclerenchyma, which provide mechanical strength to the stem. And next, uh, next part is cortex. And the cortex is differentiated into outer single layer of compactly arranged cells. You can see here, this is outer part of cortex, and this is inner part. And this outer part, outer single layer of compactly arranged elongated palisade cells, right? These are palisade cells, and consisting These palisade cells consisting abundant chlorophyll and are thus capable of photosynthesis. Then inner zone. Inner zone is made up of loosely arranged cells containing fewer chloroplasts. Cortex is then delimited by a single layered endodermis. You can see here. This is endodermis. Then vascular bundles. Vascular bundles are open and dark. Center of the pith. This is pith, and center of the pith consists of thick-walled teniferous cells. These are teniferous cells. Xylem consists of tracheid, vessel, and xylem parenchyma. And phloem consists of sieve cells, phloem parenchyma, and albuminous cells. The internode of the stem of Ephedra, this part is internode. Internode of Ephedra are quite elongated, and this is due to the activity of an intercalary meristem or meristematic plate, which is present just above the node. Stem of Ephedra undergoes secondary growth in a usual manner. And this, you can see here, this is transverse section of old stem. And beginning of the secondary growth is marked by the differentiation of interfascicular cambium, and this joins with the intrafascicular cambium to form a ring. Now, what is this interfascicular cambium or intrafascicular cambium? We have already discussed in our previous lectures. Then this cambial ring cuts off secondary xylem toward the inner side and secondary phloem toward the outer side. So you can see here annual rings. These are visible, and with each year of the secondary growth, these are growth rings. You can see here in this diagram. Then now look at this diagram. This part shows early and late wood. Now early wood, you can see here in early wood, large vessels are present, and in the late wood is composed of mainly tracheate. Then look at this third. This is a part of stem cut in radial longitudinal section, RLS, and showing large vessel, this part, and many tracheates. This part, D diagram, this portion of the stem cut in tangential longitudinal section, TLS, and showing two wide multiseriate medullary rays and many tracheates. Tracheates shows bordered pit. I can see here showing bordered pits. And in the case of Ephedra, vessels are the prominent feature of the stem. You can see here these circles. These are vessels. So this is a most prominent feature of the Ephedra stem. Now just look at these diagrams. Here, this part showing the comparison between the young stem and the old stem. Look at first at the young stem. Young stem is green and photosynthetic, as I already told you, due to the presence of chloroplast in the palisade-like cells in the cortex. So this is circular in outline. Epidermis with stomata. You can see here, this is epidermis. It's with stomata, and cortex is well developed, composed of parenchymatous cells. Then vascular bundles in young stem, they are arranged in a ring-like pattern with phloem on the outer side and the xylem toward the inner side, and this is separated by a cambium. Then pith, and pith is large, circular, thin-walled pith is present in center of the stem. Then medullary rays. Medullary rays in young stem are uniseriate, and medullary rays actually connect the phloem and xylem and they are uniciliate in young stem. Now look at the old stem. Old stem is woody and hard, then irregular in outline, and epidermis in the young stem. Now in the old stem, this epidermis is replaced by peritoneum. So now we'll use the term peritoneum for this. Then sclerenchyma tissues may be present in the cortex, and they provide strength to the stem. Then some parenchyma cells may be lignified. Then medullary rays. Medullary rays become multiciliate. It may be by the longitudinal division of the ray cells or by fusion of uniciliate rays. And you can see here these circle-like structures, they are the vessels. And this is the most prominent feature of the old stem of Ephedra. Next is root. Root of Ephedra shows typical root structure in morphology. You can see here, these are the roots of Ephedra. It shows typical root structure in morphology as well as in anatomy. The outermost layer, this is transverse section of root. Outermost layer is epidermis, and which is followed by a well-developed cortex. And the cortex, you can see here, differentiated into two regions, in compact outer region made up of thick walled cells, and the inner region composed of loosely arranged cells and their thin walled cells. And mucilaginous canals are distributed in the cortex. Cortex is limited by a single layered thick walled epidermis, endodermis. So this is endodermis, followed by single layered parenchymatous pericycle. Then there is a small diastele with the exact protoxylem elements, and the pith is small but prominent, and the root undergoes secondary growth in a usual manner. Next is leaf of Ephedra. You can see here leaves. Leaves are connate and are fused into a basal sheath, and leaves are small, scaly, and thin. They are green when young but turns brown at maturity and eventually shed. Now, SST buds rise below and at the base of the axillary shoots. Since the leaves are scaly and reduced, the carbon assimilation is done through the stem, which is green, young stem. So, in a transverse section, just look at this diagram. In transverse section, leaves exhibit an oval outline, and epidermis bear thick cuticle, and few deeply situated sunken stomata are present that interrupt the epidermis. Epidermis is followed by 2 or 3 layers of palisade cells containing chlorophylls, and the remaining space is filled with uh, spongy cells which are devoid of chloroplast, and two small these vascular bundles. They are both collateral, means these bundles are arranged in a way that they are both collateral and endark, meaning the xylem is toward the upper or adaxial surface. This is xylem, and phloem is toward the lower or abaxial surface with the xylem. And so the vascular bundles are also conjointed, meaning the xylem and phloem are closely associated and open, indicating the presence of cambium layer between them. Thank you.